Hearing about a new virus outbreak is certainly going to make us a little anxious, right? Well, we have witnessed one pandemic and we don't want another one. But unfortunately for us, there are so many viruses out there circulating in the wild that pose potential threat, and we don't even have vaccines for most of them. For example, I work on this virus called Lassa virus, and this virus is currently restricted to parts of Africa. But by 2070, it is expected to spread by at least 5 times. Now you see, when a virus infects our cells, there are proteins present on the surface of the virus called fusion proteins that recognize and bind to receptors present on our cells. And when that binding happens, this fusion protein undergoes extensive structural changes that will essentially pull the virus membrane close to our cells, allow the virus to enter cells and establish infection. But because these fusion proteins are also exposed on the surface of the virus, our immune system sees it as an important target. Our body in turn produces antibodies, which are also proteins by the way, and these antibodies bind to the fusion proteins, preventing them from engaging the host cell receptors, thus preventing infection. Now that is also the goal behind vaccination. In recombinant vaccines, you introduce just the fusion protein into your body, allow the body to mount an immune response, generate antibodies, and have them available for when an actual infection takes place. But for this, you need to design a stable fusion protein construct that is not just harmless to your body, but one that can mount a potent immune response. Traditionally, the way scientists do this is by solving for the structure of fusion protein, which obviously is hard, and then they sit and make changes to it one by one, test and validate them. This, needless to say, is time-consuming, tedious, and most importantly, it's expensive. This is where my research comes in play, as we aim to develop a large-scale platform where you don't rely on any structural information, but at the same time you can make and test thousands of different types of changes to this protein in different combinations, and then look for the desired biophysical and biochemical properties. This is not just an efficient approach, but it is also important because a virus that may be harmless today can be a threat tomorrow. And when that happens, we need to come up with quick and effective solution. That is why I believe that my work would help progress the field of virology and vaccine development in general.